Erin, can you talk us through, how those partnerships and what role do they play in bringing regenerative medicine to patients, especially, in the context of what we're going to be talking about today?
Yeah, I mean, I think, this is a new and emerging field, and so it's really rapidly evolving. Collaboration is really key to accelerating our ability to bring, potentially transformative therapies to patients. You may have certain skills, and a collaborator halfway around the world may have a complementary approach. And so the collaboration really helps us bring the best products forward, right? From anywhere from collaborations regarding automation. We actually have a collaboration right now with Yaskawa Electric in Japan for the development of a humanoid like robot to help with standardization and streamlining of manufacturing. Other partnerships, can help with understanding disease biology, partnering with clinicians or with hospitals for access to clinical samples or the evaluation of real world data. That can really help inform how best to approach various types of treatment. And then also, I think the other aspect of collaboration, which is really important, is consortia or industry working groups that can really help us learn from one another and to help with standardization across an emerging regulatory landscape. So that crosstalk and communication and collaboration can only help propel the field forward.
Yes, thank you. And Julie, I'd like to offer you the same question. And you're, as a founding member of ISCT, and we talked about before today's panel, that you're fresh off of the ISCT meeting. Certainly a great context and conference for collaboration and partnership. From your perspective as part of the Mayo Clinic as CTO, and then even with ISCT perhaps, what do the role of partnerships really mean to bring regenerative medicines to patients?
Yeah, thank you. Partnership is key, just echoing kind of what Erin had said, but as Mayo Clinic, the needs of the patients come first. And when we think about that, we look at the end in mind from the center. And how do we get to the end? We have all of these innovative research and development projects that come from either clinical or principal investigators, and how do we get them to patients and treat and cure? And it's really these relationships. So this opportunity to be able to work with industry as we're moving things forward and we're translating. It might be, them licensing the technology and we're working together to be able to execute, and then they'd commercialize it. But it's helped support our research and innovation. In some cases, it might bring in funding and resources to be able to accelerate that as we get to later phase clinical trials. It's quite expensive. I would say policy development and advocacy is really important with government agencies, the regulatory support, and then workforce development. From the ISCT hat that I wear, I will say it's interesting because it was founded 30 years ago, and it was an academic organization. Now it's really merged to at least 50% industry and academia, and then our government colleagues as well, and that really helps facilitate translation. So it's an incredible mix. We need to have these types of partnerships to be able to accelerate what we're doing in the clinic. So very productive, I would say, as we all get in the room in these different meetings to be able to share ideas. And as Erin said, have these relationships with companies that help to facilitate. We might see an enabling tool or a certain technology that we can bring in with the technology that we have to be able to accelerate that to patients.
Yeah, sure. Thank you. I want to talk a little bit about how, and Julia, perhaps we can continue on with the conversation. How are you seeing iPSC-derived therapies differentiate themselves from other regenerative platforms? So, for example, MSCs, ESCs, in terms of clinical outcomes or perhaps therapeutic versatility?
Yeah, no, thank you. It's amazing to see the wide-ranging therapeutic potential applications for the iPSCs. We've watched MSCs grow, and I'm super excited that we were able to see commercialization with Mesoblast of the technology, because there's been a lot of work in the field, and we certainly know they're safe. And I think in certain applications you will be able to see efficacy. So it was exciting to see that, but there's a lot less differentiation with those cells. So there's only certain applications that they would be used for. There's a certain degree of immunomodulatory properties. With embryonic stem cells, we're seeing a lot less and less of the research in that space. It could be ethical considerations or being able to source those cells, but I really see iPSCs growing as we have this opportunity to, for a lot of different things, as we've already discussed, regenerating tissues and organs. They can differentiate into any cell in the body. They don't really have ethical concerns. You can get them from pretty much anywhere in the skin, or you can harvest them from blood-derived cells. So there's a lot of opportunities to be able to produce these cells. So I think it makes things, it's more rapid. There's a lot of also input right now on manufacturing and scalability of these types of cells. There's publications coming out. We at ISCT, in our work group called Emerging Regenerative Medicine Technology, which focuses on iPSC, has published a paper on, what does it look like to manufacture these cells up to a master cell bank, and then how do you scale these? And the third paper we're working on is really being able to focus on the allogeneic. So I just think that there's a lot of opportunities, and there's a lot of interest, as I said before, from investors as well to look at these technologies.
Excellent. And I'm so glad you brought up manufacturing and scalability, because we will get to that in just a little bit during our conversation. So we'll jump right back into that shortly. Before we do, Erin, I would like to hear from you and your perspective, Astellas' perspective. How are you seeing the iPSC-derived therapies differentiating themselves in terms of clinical outcomes and or therapeutic versatility?
Yeah, I think to quote a commercial from a long time ago, you've come a long way, baby. These cells, I remember back in 2006, I was down in Argentina teaching a lab-based stem cell course when the original publication on iPS technology came out. And since that time, the quality of iPS cells has greatly improved. There's the utility and the fact that they're pluripotency. I think folks regard them as on par with the pluripotency of ES cells, right? Their quality has really drastically improved with various reprogramming methods, non-integrating methods. And because you can derive them from so many different somatic cell types, they really are more accessible, and many groups and companies have been able to generate compliant lines. And so I think that they're really the way of the future. Certainly, they don't have the ethical considerations that some of the original ES lines did as far as sourcing, and so it really is democratizing the use of iPS cells for so many different applications. One other thing that I think is a great or potentially a really unique advantage of them is that you can, in many situations, and I think there's some groups that have shown this, is to leverage the epigenetic memory of the starting material, and that sometimes can help improve differentiation protocols. And so if you start with a T cell to make an iPS cell and then turn it back into a T cell, you may have the benefit of leveraging that epigenetic memory to help with that lineage-specific differentiation. So I think they're just a great starting material due to these various attributes.
Can I just add to that I think that that's a great comment, because as we talk about the gene editing and someone's editing 4 and 5 genes, is there an opportunity to bring these other opportunities, such as the epigenetic history or other types of HLA matching or so forth, to be able to decrease the number of gene edits? So I think that that's really an expensive, really a opportunity for that space to be able to have something that's not going to be rejected, that you're not going to need immune suppression, but maybe decrease the number of gene edits that you need for that cell.